Let's talk about the coronavirus outbreak here in Africa. How do you get people to regularly wash their hands? The majority of Africans work in the informal sector.
People are dying and suffering.
More importantly, how do you get a vibrant and dynamic continent to stop in its tracks? Welcome to The Breakdown, BBC Africa's COVID-19 special program, helping you make sense of the crisis.
This broadcast is made possible by the Capricorn Group. This lesson is provided by Eduvision with the support of
So I want you to take out this worksheet and I want you really to concentrate on having these answers. Make sure that you can remember the order, the sequence of the process of aerobic respiration. What is aerobic respiration? It is the breakdown of an energy-rich molecule like glucose in the presence of oxygen to provide energy, carbon dioxide and water. We are going to look at the stepwise. And yes, the first process is glycolysis. The moment when you see the words lysis, we know it is the breaking down of something. It is a chemical reaction that is taking place. Of course, in order to break down the glucose into 2 pyruvate molecules, 6 carbon molecule is now broken down into 2 carbon molecules. It needs to invest 2 ATPs, so it needs energy to be released in order for this process to take place. So 2 adenosine triphosphates will release their energy, and that brings us back to 2 ADP plus P, a molecule that can be used to build up energy again. Now, during the next phase, where that glucose breaks down into 2 pyruvate molecules, it also produces 4 ATPs in order to capture the energy. And there we see the 2 pyruvate molecules. It is important for higher level to know glucose, 6 carbons, breaks down into 2 pyruvate molecules. It first needs 2 ATPs and then it produces 4 ATPs. It also captures the hydrogens, and the hydrogens is now carried by 2 hydrogen carriers, and that brings us to the fact that NAD adds a hydrogen, and now I have NADH. NADH will only be the molecule that is transporting the hydrogens to the next phase. Glycolysis is outside the mitochondria in the cytoplasm. So we have a yield products that formed during glycolysis was 2 3-carbon pyruvate molecules, 2 ATPs. 4 was produced, but 2 were invested. And then we have the NADH, the hydrogen carriers that captured the hydrogens released by the glycolysis process. This part of the reaction was not depending on oxygen. Oxygen, when oxygen is present, we have aerobic respiration. 2 pyruvate molecules are transported now from this process through the mitochondrial membrane to the matrix. And you do not have to know in which molecules it is converted. But we move now to the Krebs cycle, and this part needs oxygen to be present. The 2 pyruvate molecules are now transported to the matrix of the mitochondria. In order for the ATP to capture the energy, to be the molecule that stores the energy, ATP is formed when an enzyme transfers a phosphate group from a substrate to ADP. Then the Krebs cycle is taking place in the mitochondrial matrix. Important, it takes 2 turns of the Krebs cycle to oxidize 1 glucose molecule. And it's in the mitochondrial matrix. See in this slide is that the Krebs cycle is a circular event that is taking place. It releases carbon dioxide from this stage. It captures all the hydrogens into a molecule NAD+, and it forms ATP. During the Krebs cycle, look at the spelling. It also needs another hydrogen carrier, which is FAD. FAD capture or transport a hydrogen, and it forms now 2 hydrogen, FADH. Of course, this happens 2 turns. Why? Because the glycolysis ends up in 2 pyruvates. Now I have 4 carbon dioxides that was released during the second turn, the NADH and the FADH brings the hydrogens and the molecules form. ATP molecules form. So the yield after 2 turns of the Krebs cycle will bring us 2 ATPs, 6 NADH, 2 FADH2 and 4 molecules of carbon dioxide, 2 per turn. Now it brings us to the last part. And before starting this part, I want you to think of holding a very warm potato in your hand. You cannot keep it in your hand, so it burns your hand. And then you throw it to this hand, you throw it to this hand, you throw it to this hand, and after a while you realize now the potato is cold enough so that you can keep it in your hand. Grade 11, I want you to understand, with that throw of the potato, the warmth, the heat of the potato is actually radiated or transported to the environment. The heat heats up the environment, the air in the environment, and the heat is not gone. Energy can never be lost. It can only be converted to another form. And that is exactly what is now happening in the inner mitochondrial membranes. There you can see what are we talking about. All the different spaces of the mitochondria. The hydrogens now moves via diffusion through ATP synthase to make ATP. All the NADH and FADH2 convert to ATP during this stage of cellular respiration. Each NADH converts 3 ATPs. So if we quickly can make a calculation, there were 6 NADH transporters coming into this phase. So 6 times 3 is 18 ATPs, just for interest's sake. And the FADH2 converts 2 ATPs each time. If we now move to the last stage, it helps us just to quickly get back to the 6-carbon molecule glucose during the glycolysis process breaks down into 2 pyruvates. During this process, 4 ATPs were produced, but 2 were used beforehand, and the hydrogens released there from the 12 hydrogens that builds up a glucose is carried to our process of oxidative phosphorylation, the 3rd stage. Then the pyruvates needs to enter for the Krebs cycle into the matrix. In the matrix, there we see 2 carbon molecules and then 4 carbon molecules during the Krebs cycle is formed. 6 carbon dioxide molecules formed. 2 ATPs are already storing energy in stage 2. And then we see that 8 NADH molecules and 2 FADH molecules need to bring the energy-rich hydrogen molecule process. So eventually there are about 38 ATP molecules formed during the process of respiration. And this slide only shows us what is taking place. When hydrogen are accepted by the oxygen, we have the formation of water. We say that hydrogen is oxidized into water because hydrogen gains an oxygen molecule. Oxygen is therefore the final acceptor of water. Phosphorylation, just to make sure that we really do understand. If we look at oxidative phosphorylation, we see steps that looks kan hierdie gebruik nie. Don't save.
This lesson is provided by Eduvision with the support of sponsors.
News just in. Brace yourselves as One Africa TV's brings you a newly
We listen.
The way they operate have to be different. Because we are losing lives here.
with over 1 million impressions on posts, we bring you
Community Voices,
Sport. Consumer Watch.
Social media commentary,
The weather. And so much more.
today on One Always There.
This lesson is provided by Eduvision with the support of sponsors.
We start with the red blood cell. Of course, we know that the red blood cell has a biconcave shape. And that biconcave shape is to increase the surface area to volume ratio. Because inside the red blood cell is a very important protein. And we call it hemoglobin. Hemoglobin has the function to transport oxygen in the form of oxy hemoglobin. If we look at the features of a red blood cell, we know it is small. How is that an adaptation for its function? It is small so it can squeeze in the tiny capillaries. It has an elastic membrane. Again, it can move. Because it's elastic, it can fold and squeeze through the tiny small capillaries in order to transport the oxygen. It contains hemoglobin. Johanna, hemoglobin is what? What type of structure is hemoglobin? Is it a carbohydrate? Is it a fat? It is a protein. Now, revision class. We have two types of protein. Protein is divided into globular and fibrous. Nah. Fibrous proteins, which is the nails, the hair, all those that need to be thread-like. Nah, fibrous. Fiber. Hair, nails, your tendons, that is fibrous proteins. Your muscles, fibrous. Can you see it? String-like, nah. And then we have the globular proteins. Okay. Now, if I say there is no what is that for, Abigail? No nucleus. How does that help? What is the adaptation? More space, more hemoglobin, more oxygen can be transported. No mitochondrion, because if there was a mitochondrion, what will happen? The mitochondrion will use the oxygen, and the oxygen will not be transported, it will be used. Now, it is like I am giving you a box of chocolates to give to your mom as a present, and you eat all up along the way. Then your mom receives no present. Nah, same, then the cells will not receive the oxygen. Good. And then we already said biconcave, surface area to volume ratio. We will remember that one. Okay. Now, I want to keep my nucleus, but you must make space for all the hemoglobin. So we understand that. Now, when we look at the Oxy Hemoglobin combination, we want to understand that oxygen is transported from an area of high oxygen tension or pressure, and that is in the lungs. Nee, we do understand that. When I breathe in, lots of air in my lungs. That makes a pressure. Inside the air that I breathe in, there is also oxygen. So we talk about the partial pressure. Oxygen is part of the air that I breathe in. And when I have a high air pressure, the oxygen inside also have a high pressure. And we know that if we move to the area of low tension, oxygen tissue. So, we know that oxyhemoglobin need to transport oxygen from an area of high tension or high partial pressure, and it must reach where the tissue are respiring. Sorry. So now we have to look at oxygen saturation. Saturation is the state of being saturated or the action of saturating. So when I have a beaker full of water and I add sugar and I add sugar and I dissolve it, I will reach a stage where it is saturated with sugar. But I want you now to think of oxygen that can also reach a stage of saturation, where it takes up a very high amount of oxygen, where hemoglobin takes up a very high percentage of oxygen. Now, this is what we are going to study today. So we know that in the alveolus, there is a high tension or a partial pressure of oxygen. That makes diffusion fast. High partial pressure, low partial pressure. When I have a high pressure, it means that the equation, the reaction between the hemoglobin, and this is a picture of hemoglobin, higher level, is a glomerular protein. Ne? You remember the structure of a protein? It starts with a primary structure, which is determined by the type of amino acids in a long string. Then we have the secondary structure where there are bonds and it starts to fold in a pleat. You remember? And then it folds again to make a three-dimensional structure. And here we clearly see a three-dimensional structure with 4 spots for the oxygen to attach. And the moment when we attach another group, an iron group in the case of hemoglobin, we have a 4th structure in a protein. Are you with me? And that is a question, I promise you, to discuss hemoglobin as according to its protein structure. Why is it important? It's important to understand that hemoglobin eventually has the iron attached to it. Ne? It gives the possibility to attach oxygen to it. Iron can attach the oxygen. How many oxygen molecules do you think will attach 2, 4, 6, 8. 8 oxygen molecules attach for diatomic, yes. But eventually I will have 8 O's, 8 atoms of oxygen, which gives me, if I have a lot of hemoglobin in one red blood cell and I have millions of red blood cells, a lot of oxygen atoms that will travel through my body. We do understand that? Okay, so that is important. Now, there I have a very high pressure. Therefore, the oxygen easily get saturated with the hemoglobin because of the high pressure. But what do I want to happen when that oxygen, highly saturated, reach the tissue cells? What do I want to happen? I want the hemoglobin, the oxy hemoglobin, to release the oxygen. Do you understand that? So I want this to attach and get saturated there. Nee. But here, I do not want the oxygen to stay bonded to the hemoglobin. I want it to get released. So how do I want the saturation here? Very low. Does it make sense? And it is perfectly, we are perfectly designed for that. Hear this. So, if I have the pressure of oxygen on the x-axis, the pressure, the partial pressure of oxygen, and I have its saturation, we set in the alveolus high pressure. When I have high pressure of oxygen, I want high saturation of hemoglobin with oxygen. And that brings me there. Do you see? When oxygen is high pressure, I have a high saturation rate. But when I want to release it at the tissue, how is the pressure of oxygen in tissue cells? Low. Why is it low? Because the oxygen was used during respiration. Nee. Do you get that? Do you get that, Karos? In the tissue, I have very low oxygen because oxygen is used up. Oxygen is changed to carbon dioxide there. Nee. And therefore, it brings me to, when I have low pressure of oxygen, I have low saturation. And isn't that exactly what we need? Do you hear me? We need to. When I have high pressure, I want high saturation, so that all the hemoglobin and the oxygen combine and form hemoglobin easily and very fast. Yes? You follow? But the moment when this oxyhemoglobin travels to the tissue, do I still want to have this strong relationship, this strong bond? No, I want them to release the oxygen. So pressure low, saturation low in hemoglobin. Easily understood. Now, good. So now, class, I want you to page 277. The question there is, describe and explain the oxygen dissociation curve. So, oxygen associates when the pressure is high. But as the oxyhemoglobin travels, I want it to dissociate. It must dissociate. It must break this bond. Nee. So, the vertical axis shows the percentage saturation of hemoglobin with oxygen. shows the amount of oxygen available in the surrounding medium. The dissociation curve, please follow in your book, because that paragraph you must